let's move on now to the last part of the syllabus, which is looking at inheritance patterns in a population. So we are now considering, can population genetic patterns be predicted with any accuracy? So we've looked at now, what is happening at a molecular level, how do we then predict it? But now we're saying, can we actually now predict not just individual genetic patterns, but like population patterns with any accuracy? So these are the techniques we use. Okay, so we use DNA sequencing, which involves firstly taking some DNA from the cells, and then you identify identification of sequential order of nucleotides. So DNA is basically made up of the deoxyribose sugar. It's made up of phosphate, and it is made up of the nucleotides that actually bond together. So when we do DNA sequencing, our DNA, our actual, what makes us unique, it's seen in the base pairing of the nucleotides. So there are 4 nucleotides, and the 4 nucleotides are basically, they're called A, which stands for adenine. There is guanine, there is cytosine, and then there is thymine. Alright, and the way they pair up is, I like to think of it, I've been, like the mnemonic that we used to use was apples in a tree and cars in a garage. Alright, so adenine, which is spelled Adenine, pairs up with thymine, Thymine. And then guanine pairs up with cytosine. So guanine pairs up with cytosine, cars in a garage. Alrighty, so when they pair up, that's like these sequences, that's what makes up our DNA. So what happens in DNA sequencing is we identify the order of the nucleotides. How have they paired? Like we identify the order of a nucleotide. So we may have, for example, A, G, C, T, T, T. That's literally what DNA is, basically, right? So we use computational processing to compare whole genomes, and we also do so we want to compare whole genomes, basically. So what we do is we get single nucleotide information. We remember that each individual has a different genome, but there are lots of similarities between every human, basically lots of similarities in our DNA, but we do have like parts of the DNA that are unique to us, obviously, otherwise we'd all be the same, right? So we obviously have that diversity. There is identification of similarities and differences, and modern computation is so good because it allows patterns to be identified, and through that we trace inheritance of genes. How have genes been inherited? We trace inheritance of alleles, as we spoke about alleles just a couple of minutes ago, and we look at SNPs. SNPs are single nucleotide polymorphisms. So this is where single SNPs, like these single nucleotide differences between people, that give us our unique structure, unique DNA. That's what single nucleotide polymorphisms are. So that's DNA sequencing. The DNA profiling. So we collect DNA samples from the cells. We then digest it. So we basically cut the DNA into small pieces. We use something called restriction enzyme to cut it at specific places. And then what we do is we do a PCR, which is like a polymerase chain reaction, and we get lots and lots of copies of the DNA. And then we put it into gel electrophoresis. This thing here, basically what happens is DNA travels from this positive end to the negative end, and as DNA travels, like you can see similarities between the benchmark that you sort of put in here and your samples here, the unique samples that you put in here. And if the bands like match, if they match, like whichever one matches the most, like that's where you can sort of see similarities. This is how fingerprints can be tested. This is how also paternity tests also happen. So this allows for a comparison of patterns. Now, the more similar they are, the more the higher the degree of relatedness. And we can observe conserved regions of the DNA. And the reason that it is effective in humans is because we have large stretches of junk DNA as well. So, not all DNA codes for proteins, a lot of it is just junk DNA. So, um, large scale projects. So, now how do we use this information to look to implement large scale processes? So basically what we do is that we look at large scale collaborative projects by firstly looking at the trends and relationships between all these different elements, and then the application. So conservation management is a really good example of it, because conservation genetics is basically fields that combines molecular genetics, ecology, and biodiversity sciences, so that they can propose species management strategies. And they use population genetics to identify which alleles specifically are at risks, and through that they then look at providing ways to manage and preserve biodiversity. So the Australasian Wildlife Genomics Group, that is run in the Sydney University. So they have projects including the Devil's Tool and Tech project, which look at the Tasmanian devils, the Koala Genome Project, the Marsupial Genomics, and they also collaborate with the Australian Museum, Chicago Zoological Society, and National Cancer Institute. So they do that in order to collect that data, research the patterns and the relationships, and then put in these collaborative projects in order to preserve these alleles that are going extinct. Inheritance of disease is also so we can also use these technologies to look at the inheritance of disease, right? So only now, something that's really important is that there is only a 0.8% nucleotide variance among humans. That means that there's a very, very little variance in humans. Very little. But obviously, it's still enough because, we are surviving in the natural environment. But it's very little. So that means that it's within that 0.8% buffer. That's where genetic disease are contained, right? That's where genetic diseases have to be contained within that variance that is between different people. So we want to understand the causes of genetic disease. We want to understand, is it hereditary, or is it rather a more random mutation? What are the alleles behaving like, right? And so after we do that, we can then design therapies to repair genetic disorders. So we can use this information to repair genetic disorders. And so there was a huge project at the beginning of the 2000s, just when computers and all of these and all the technology was on the rise, and so they collated data on thousands of genomes, and it was a huge scale process, and it was used to identify genes and variable regions. And through that research, actually, they identified the genetic causes for breast cancer, for Alzheimer's, for colon cancer as well. So we know now that it's within that variance buffer where the genetic diseases are. So we can use this data to actually identify trends and relationships, and then implement therapies and projects to stop genetic diseases, to contain genetic diseases. Finally, human evolution. So cultural regional groups are often linked by prevalence of particular haplotypes, by particular sort of traits, right? So mapping haplotypes allows us to trace the movement and evolution, and through this, actually, they were able to trace back to common ancestor, and the common ancestor that we've been traced back like evolution back to, is mitochondrial Eve. Now, mitochondrial Eve is because everyone, you have your mother's mitochondria, so it's it goes back like the maternal line. That's why it's mitochondrial Eve. It's a mitochondrial Eve lived in Africa 20,000 years ago, and this was done. And another project that is currently in place is the International HapMap project. So that uses human genome data to identify SNPs, haplotypes, and haplogroups. So from this information, we are able to trace back lineages between populations. So Any questions so far?